Okay, let's have a class and let's continue to talk about slides, lecture 1, then we'll move on to lecture 2, talk about a microscope. So we mentioned Louis Pasteur about a week ago, and the swam neck flask, which is repels the spontaneous reaction in the microbiology level. Because of this, we call it a swam neck, looks like, okay? And then people did the research, but they find there's different results. Some people find even the boiled water still have bacteria. So John Tidell and Ferdinand Cohn verify that it's possible if it's heat-resistant bacteria or the bacteria has endospore. We're going to talk about endospore real quick, pretty much on Tuesday. We'll talk about the endospore structure, what it looks like. Because I have get some question from some of you, and that's very good. You're interested about this new material. Now, by the same time, there is another great scientist, microbiologist, Bob Koch. He's from Germany, and he did lots of research. The most important thing is that he's talking about the Koch's postulate, which is very clear to establish the relationship between a particular microorganism and corresponding to a particular disease. So this comes out with a very important the postulate. So what is a postulate really talk about? So the number one, what they say is healthy body. Okay, I'll just say healthy body is absent from microorganism. So which means if you're healthy people, you shouldn't have tuberculosis unless you get sick from tuberculosis. Okay, then the second one is that the target bacteria can be grown into pure culture. And we're going to talk about this pure culture real quick. What is that? And the third one, which you can look at here, the same disease will happen if the pure culture infected a healthy body. So for example, if you have a tuberculosis, the tuberculosis you got isolated to get a pure culture. You used to inoculate it, or we say spiked, or infected a healthy body. Then the same tuberculosis will be happen. And the last one is re-isolation. So what means re-isolation? Which means the same microorganisms could be isolated again from the diseased host. So this is 4 items of Koch postulate, which is well explains the relationship between a bacterial disease, a bacterial strain, and the body, the human body. But I want to be cautiously to say this theory developed about 200 years ago. By the time the immunology has not been developed. So if you look at the first one, the microorganism must be present in every case of the disease, but absent from healthy organisms. If you read that today, it may not be true. So for example, people have coronavirus symptoms. They obviously contaminated with coronavirus, but there are lots of times there's asymptoms happen. Is that right? Even people which is contaminated or infected with some pathogen does not mean they were showing the symptoms, because of immunology. So the immune response. So this is not included here, because by the time it's not been developed. And the picture here which is using is a tuberculosis. So microbacterial tuberculosis, we'll talk about detail next week. Isolated from the TB patients, and then we get the pure culture, and the pure culture go to a guinea pig, and the guinea pig get the same tuberculosis, and the microbacterial tuberculosis can be re-isolated from the guinea pig. That is Koch postulate. And Koch did a lot of the other things. So here is the thing, a pure culture. So what is pure culture? Which means there's only one genome name, one species name, and there's no contaminants. So the first threshold, or we say the difficulty to get the pure culture, is what kind of the media you should use. What kind of the bacterial media you should use. By the time, there are two different theories. From Louis Pasteur group, they are doing serial dilution on tubes. This is you learned in chemistry class. So I have a bacteria, chunk of bacteria here. I'm preparing a lot of tube. Okay? All these tubes, let's say, containing 9.0 ml. And I'm adding 1.0, go to the first one. Then we mix very well, go to second one, go to third one, go to the final one. And we ideally want the last one has one single cell. Is that going to work? No. Because theoretically possible, practically, no. We're never going to verify there is a one single cell there. So, using liquid media certainly does not work. Therefore, Bob Koch's group, they're trying to use solid media. But the question is, what kind of the material the solid media can be using? The challenge is that the bacteria can grow, but the bacteria cannot eat it. So they tried lots of material. Let's say they tried the jelly. Like the grape jelly, you put in the middle of your hamburger or the bread. What they find? Yes, the bacteria growing there, but the jelly is also gone because bacteria use the jelly. So what they do? They spend a huge time. And by the time, Walter Hesse is one of the research scientists at Koch's group. He's spending lots of time to do. In 80, I believe in 80-70s or 80-80s, in a very hot noon at Berlin, his wife, Fanny Hesse, that's Fanny Hesse, prepared the lunch. He said, honey, it's a time for lunch. And today we are eating French pudding. Then what has he think about the pudding? That's a good idea. What is that? It looks like agar. The material is polysaccharides coming from seaweeds. That is a really good material. Why? The agar, they will be melted at 50 degrees Celsius, solidified at room temperature, or let's say 30 degrees Celsius. It's very like a pudding. The French pudding looks like that. So, Walter Hesse told Bob Koch the idea from Fanny Hesse, and Bob Koch said, yes, it's a good idea. Then they started to using agar to isolate the bacteria. Now, by the time, what is the container for the agar? Richard Petrie, another scientist in Bob Koch's group, find a dish can use as a container to hold on agar. That's why today we call Petri dish, coming from Richard Petrie. So, how we get a single colony? This is how we got it. You have a solution, you have agar. You do a very intensive first charge streak using loop. And then you flame the loop from the edge of the first one to a less intensive. And then you flame the loop again from the edge of the second one to a much less incentive streak. And then you flame the loop again to a final one. And these plates need to be incubated 35 degrees Celsius for 24 hours. What we will see, you will see like that, these dots. Look at those dots. And ideally will be looks like here. Look at that. These are single colony. This single colony represents one genus name and one species name. That represent a pure culture. And then you see these colony. This colony is a cluster of bacteria cells. How many are there? That's huge. More than 10 to the power of 6 to 10 to the power of 8 cells. That's called a colony. Keep in mind, that is not one single cell. It's a cluster of bacterial cells. A group of bacterial cells coming from one mother cell, representing one genus name and one species name, that is called a pure culture. It is very important we have that. And we will get practice to get the pure culture in the second to last class of this semester. Because we don't have a lab, but I do want everybody to get a feeling, a training. What is the pure culture? What is the agar? Okay? That is very important. So here, I show you something regarding the streak plating. So this is they did the streak arch, and this is they also did the streak arch. You see the single colony. And these colonies are important. So for example, sometimes you see the yellow colony. That could be Micrococcus laelius, which is coming from the soil, but it's contaminated food products. If you see a pushable colony, that might be gonorrhea. It could be Neisseria gonorrhea. So those characteristics of the colony sometimes will tell us some of the information. It's a presumptive identification. Now, the detailed identification, if we want to know what the species name, for example, if we want to know is that an E. coli, is that a salmonella, we need to do a biochemistry test, we need to do a genetics test, or we may need to do a whole genome sequencing to know the sequence, like ATGC of the bacterial culture. But all these coming from a single colony. So, Louis Pasteur did a serial dilution, tried to do serial dilution on liquid media. Bob Koch's group, using agar, get a single colony. These methods, right here, we say serial dilution on solid agar media. Because we are diluted a cluster finally from here, the heavy smear, to a single colony. So this is a serial dilution on agar plates, and we call it streak plating. Okay, Last one. Name of classifying microbes. We using binomial nomenclature. So every bacteria, we have two sections. Let's say the easiest one, E. coli. And I make jokes all the time. I said, you even failed with microbiology class, but we will never forget a bacteria's name even after 30 years. That is E. coli. So what is that? It's Escherichia coli. That is E. coli. You have to write italic. What is this Escherichia? Genus name. What is coli? Species name. I believe Escherichia is from a scientist name from Japan. Coli is a morphology of the bacteria. Looks like a large shape. So we have other examples. Bacillus arsenicis, Clostridium botulinum. Clostridium is a genus name. Botulinum is a species name. Streptococcus pyogenes. This one we mentioned, called strep throat. Streptococcus is a genus name. Pyogenes is a species name. But there is one exception. What is this exception? Salmonella. If you look at the textbook, you look at the paper, lots of people write salmonella is italic, but let's say Thompson. This is normal, non-italic. Why? Because salmonella have 6 subgroups. So salmonella, if you want to write it correctly, is salmonella enterica subspecies enterica Thompson. All these are italic, and this is not. And people think you are very smart, so what they do, they skip the whole section. So when you see Salmonella Thompson, no italic. That's always a tricky question for a PhD qualifying exam. So the PhD students need to get a degree, they need to go through the qualifying exam. That's always a question. Why Salmonella Thompson is not italic? Because the correct answer and the easiest answer is Thompson is a subspecies name, is not a species name. It's very interesting, but it's a very tricky question. This is a subspecies name. Okay? That's very important. Okay, we finished this section. Let's go to lecture number 2. Okay, this one we want to talk about is a microscope. Because of the snow and the weather, we have to catch up with the progress of the class. So I'm going to be a little bit. Some section over here will be very concise. Okay, we don't want to go detail of these. Otherwise we're never going to finish these materials. So we want to be a little concise. So, okay, microscope. So we already talked about what is microorganism. A microorganism is too small to see by naked eye. So we need a tool. A tool is called a microscope. And I ask my students to grab a microscope here. Some of you know, some of you never touched it. That's a microscope. So we're going to talk about these things. Microscope has lots of different categories. Bright field microscope, dark field, phase contrast fluorescence, and confocal laser scanning microscope. These are we call light microscope, which is light is an energy resource. If we're using electron as an energy resource, we got two different type of the electron microscope. Is transmission electron microscope and scanning electron microscope. We will be talk a little bit of detail about bright field microscope, which is belonging to light microscope right here. For all the others, we will be very brief. Okay, this is microscope. Microscope, we already mentioned, because microorganisms are very small. They measure in nanometers to the largest protists using the bacteria would be about 200 micrometers. It's very small. And this is common units of the measurements. You can use in this table if you need it. Okay. So we want to talk about something, but we're going to talk about here first, the structure. Okay. Let's go here. Bright field microscope. So first question, what is the most important part of a microscope? It's always the lens. This whole thing may be $2,000. The lens could be $500 to $1,000 because it's the most valuable part. And there are two lenses. What are the two lenses? The first one is using eye close to it, and the second one is close to the objective. Therefore, we have objective lens and ocular lens. And this gives us the first question, first concept. What do we call total magnification? The total magnification is the magnification of objective lens right here, multiplied by ocular lens. And the ocular lens always is 10x. Objective lens could change. So, if we are using this guy, the very short one is 4x. This is 10x. Then you have a 40x, and the last one is 100x. Okay, we can switch it. It is a very interesting material. So that could be 100x, that could be 40x. So what is the total magnification? Could be 1,000, because 100 multiplied by 10 equals 1,000. If the objective lens is 40x, that equals 400. So that's the first one, total magnification. The second one. It's very interesting. Look at these lenses. What do they look like? Did you notice that? I just showed you. This is stage. The distance between the lens to the stage is decreasing with the increasing of the magnification of ocular lens. 4x, 10x, 40x, and 100x. And this distance we call working distance. And the energy resource is right here. So you can see it very clear. 4x small. Is that right? That is very small. Little large. Large. The largest one. Okay. Now next one we want to talk about something physics. So we go back. We go back to here. Okay. So the light going through here. Light going through here. Light going through here. Going through here. Going through here. Light going through here, going through here, going through here. Do you notice there are two media here? One is air, another one is what? It's glass. So when the basic physics knowledge, when the light going through air to a glass, what will happen? It will bend it, called refraction. You don't have to worry about the slides, it just explained to you. But basic knowledge will be happen refraction. So I believe you did those tiny experiments in the elementary school, high school. You have a bottle of water, you put a pencil there, and then you watch from the side, you're feeling the pencil is bended. That's called refraction. And the refraction will be increased when you change from 4x to 100x. So, what happened? If you use the eyes to go here, change from 4x to 100x, let's just do a demo. You are feeling fuzzy. You're not going to do a clear observation because the refraction is increased. So what we do? Put oil there. So we're going to put a large drop of the oil. We call oil immersion. Why? Because the refraction index of glass equals refraction index of oil equals 1.5. Larger than the refraction index of air equals 1.0. So that's why when we do the observation of bacteria use a microscope, we need to put the oil there. Because once we put the oil, the glass air is gone, and the glass of the lens is the whole system. This is the whole system. So what's the function of oil immersion? Remove air gap. That is a very important characteristics, and we can say action. We do the observation use light field microscope for the bacteria, which is called oil immersion. A big drop of the oil in the middle. Okay, that's oil immersion. Now, what else we want to talk? It's very interesting is we want to talk about resolution, which means how we going to differentiate two major closely attached particles. So how we usually do? I don't know whether we have a plug-in here. I may not have it here. May not have it here. Okay, it's only demonstration. So what's that plug-in?
There's a plug on the side here.
Oh yeah. Sure. Let's see if this works. Because it's expensive. No, I'm not going to catch it. I'll just do a demonstration. So once you plug in, okay, we're going to move the side all the way to the end. And then make sure the light going through the condenser at maximum amount. So why we do that? This gives you another one called the resolution power, or P. What happened? Maybe touch the something. Why the projector? Never mind. Well, I can explain to you. You can look at the slides. Okay. I'll just use this to teach. The whole thing we can talk about. You can look at your slides, we can talk. So, if we plugged in, we're going to talk about the resolution power. This is a resolution power. I teach most of the classes to use slides, to be honest. I just, because this is general education, we just want everybody from different background to be knows. That's why using slides. So I usually don't use it. I don't want to try to figure out this technique. So the resolution power. What is resolution power? Z equals lambda divided by 2 and NA. So what is lambda? These type of the microscope, we call it a bright field light microscope. If you plug it in, you press a button and this edge you turn all the way to the end. Let me see whether I can. Okay, I can try. I find it. I can try to see. Okay. Okay, works. If see the light, if I turn all the way to the end, that's called the voltage control. What is the wavelength? 625 nanometer. Now what is NA? This is numerical aperture. Where it is? When you look at the microscope, you see the edge. There is like 100x OU 1.25. So if you look at the lens will show you 100x OU, which means OU immersion 1.25. That number is numerical aperture. What is that? We don't want to talk about too complicated. This is like size of the microscope engine. So if you want to do a calculation, what it equals? 624 nanometer divided by 2, 1.2 high. This is equals 260 nanometer equals 0.26 micrometer. What is this number important? What is the average size of a bacteria? Let's say Staphylococcus aureus. The average size is about 0.5 micrometer. And the resolution power of this light microscope is 0.26 micrometer. What this means? Which means you can use a light microscope to do the observation because this is larger than the resolution power. But how about virus? What's the size of the virus? 0.01 micrometer. This is less than 0.26 micrometer. Therefore, can we use light field microscope like this to do the observation of a virus? No, we cannot do it because the resolution power is not good enough or strong enough. So, in order to do the observation of virus, what we need to use electron microscope. So that's a very common knowledge. Okay? So that's basically what we have is for the introduction of the light field microscope. Okay, now let's talk about dark field microscope. You can go to look at your slides if you have a computer or have you printed out stuff. So what is dark field microscope? We can draw here. Okay. We have light. We have a stage. We have this lens. So what happened? What's a dark field microscope? Dark field microscope. Underneath the condenser somehow like here, there is a solid block. When the block is there, the light going through condenser, going through stage to the lens, will become perpendicular, 90 degrees. So, what happened for the dark field microscope? The background is dark, and your specimen, which means your sample, is bright. Okay, why this is important? In the clinical area, the dark field microscope used to identify 3 major bacteria and a very special bacteria we call spiral sheets. And you heard some of that. Number 1, Treponemian pallidum, which is on your slides. This is the one called syphilis. What is syphilis? Number 1 sexual transmitted disease pathogen in the world. Number 2, Borrelia burgdorferi. What is that? The pathogen causes Lyme disease. What is Lyme disease? People do deep ocean fishing or activities. You will see the target rash on the arms. That's Lyme disease. Number 3 is Leptospirosis interrogans. This will be a very interesting hook shape, like an S hook shape. Those 3 are called spiral sheets. It's a very special bacteria. Their major components is asexual filaments. It's a kind of like a protein. It's very soft. So they cannot do a staining. You know we can use gram stain to identify bacteria, gram positive or gram negative. These are very soft materials they're using. If you use heat, they will be melted. So we have to use a very special microscope, which is called dark field microscope, to do the observation. But the key thing is this block. Okay? So that's dark field microscope. In your slides, you can see a Treponema pallidum. Okay, phase contrast microscope. This is something we hope you understand, but we usually using draw pictures to let you understand. The phase contrast microscope, because there's a little bit of physics behind it. We talk about number 3 is phase contrast microscope. So what is that? We're going to talk a little bit about physics, about the wavelengths. Okay, if the wavelength is like this, there is another wavelength. If it's like this, what will happen? It will become very light because 2 peaks overlapped. if I have a wavelength, it's like this. What will happen? It will become dark because the peak and the bottom, they interacted. Okay, this is your stage. This is the place you can see. Okay, this is what normal happens. We're not going to see anything. Let's say, assuming. How they do? They put a phase ring around here. They put a phase ring. The phase ring will change the wavelength speed. Change light wavelength speed about 1/4, either ahead or backwards. Then what's going to happen? The curve may be shifted like this. Then you can see it. That's a principle, a easiest principle. So normally, based on the wavelengths, you cannot see it, because could be peak and the bottom overlapped. And now I have a phase ring. We're going to change the wavelength speed. It could be 1/4 ahead or backwards. Then what happens? It will be shown up, because the wavelength is not interrupted in anymore. Now, why phase contrast microscope is important? You can see the internal or inside the structure of some of the bacteria, especially eukaryotes, yeast, or molds. Because we get to know the inside structure, we will know what happened for the microorganism. That's why use phase contrast microscope. The important thing is this phase ring. Okay, our slides explain that very detailed, but you don't have to go that detailed. It's a physics behind it, but I just explained to you the important thing is this phase ring. And we have a picture talk about that. And we also have a slide which is compare the bright field phase contrast and dark field microscope, what it looks like. Okay, the next one, fluorescence microscope. Fluorescence microscope are a little bit different. Why? Fluorescence microscope. So what is fluorescence? Fluorescence is light yellow. Some bacteria has natural fluorescence. For example, a very special, common sporage bacteria called Pseudomonas fluorescens. The species name is fluorescence, so they have a fluorescence like light yellow color. It's a sporage bacteria. It's not a pathogen. Fluorescence microscope, the difference compared to dark field, phase contrast, and the light microscope is the energy resource is UV light or blue light. What this means? This gives you a very shorter wavelength. So, based on this equation, what you get? If this lambda is go down, the resolution power value is go down, but the resolution capability is increased. So you understand the story behind it? We want the wavelength is less, because when it's go down, the resolution power value down, but the capability is increased. So that's why we use UV light or blue light. It's the first thing for the fluorescence microscope. Second thing, when we're using fluorescence microscope, we're usually using a staining. The stain we call it demi stain. This stain is very special, but it's very important. It is basically red is dead cell. I don't have green color. Green is live cell. Because the demi stain is staining DNA, sometimes RNA, is staining the nucleic acids. So we will know whether the bacteria is live or dead. And that is important. Especially we are treating them using heat, or we may be using antimicrobials, because we want to know whether the bacteria is dead or not. If it's red, the dead cell. If it's green, the live cell. But if it's injured cell, what happens? Usually it is brownish color. It is injured cell. So that's what fluorescence microscope looks like. Okay, how about last one? Laser confocal microscope. This one is special. So I want to say the laser confocal microscope is special. Why? Fluorescence microscope, dark field, bright field, or phase contrast is still light. So that's relatively cheap. Laser confocal scanning microscope. This is expensive. A microscope usually 25 to 50K because of the laser. In the United States, a German company is created those material and sending to the United States called ZEISS. They are create different type of the microscope. The energy